So Ali is, as I said, midway through his PhD, and you can see up there I recruit John Bowman and Rolf Nielsen into the micro area.
Yep. Okay, thanks, Jim. And good afternoon, everyone. Thank you for coming. Today I'm gonna I'll talk about part of my PhD project, which is my first chapter, which is looking at lactose starvation response of Lactobacillus casei strain GCRL163. These strains considered as probiotics, and it is originally isolated from the cheese. So, what's the probiotics? The Food and Agriculture Organization and World Health Organization define probiotics as live microorganisms which, when administered in a equal amount, confirm the healthy benefit to the host. So, what kind of benefit they can bring to the host? It has the ability to enhance the immune system by producing some component can enhance or stimulate the immune cells. Also has the ability to inhibit the pathogens growth and adhesion. Plus, has the ability to metabolize some bile acids and producing some short-chain fatty acids. So the common way to deliver this kind of microorganisms to the human is using a fermented product like fermented milk, yogurt, and cheese. And since my strain is originally isolated from the cheese, so today I'm going to focus on these microorganisms from the cheese, like Lactobacillus plantarum, Lactobacillus lactis, casei, and some bifidobacterium called bifidobacterium longum. And basically the probiotics divide to two groups in the cheese. They call it the first group called lactic acid bacteria, which contribute to the acid production during the cheese maturation, and non-starter lactic acid bacteria, which generally play significant role during the cheese, significant role for the cheese flavor and the texture. And my strain is Lactobacillus casei, which is considered as non-starter lactic acid bacteria. Still a group of these microorganisms of Lactobacillus is commonly used, as I mentioned before, in the fermented product, and the cheese one of it. And the cheese during the processing is imposed several kind of stress on these microorganisms, like acid stress, osmotic stress, and nutrition stress. And it will be well documented that the cheese lacking lactose as a major carbon source for these microorganisms to grow. So Lactobacillus must be able to withstand in this harsh environment condition during the cheese maturation. So to date, there's a few reports investigate the physiology response of this microorganism, which is non-starter lactic acid bacteria, particularly Lactobacillus casei, in this environment. Therefore, we study the effect of nutrition deprivation in Lactobacillus casei strain GCR163 harvested in the stationary phase from modified mMRS with different level of lactose. We use two analysis way to look at the protein suppression. We call it one-dimensional one-dimensional dodonium sulfate polyacrylamide gel electrophoresis, or we call it SDS page, and 1D LC-MS analysis. I will go in details about this technique later. So we hypothesized that this strain with GCR163 can find alternative source of carbon during lactose starvation, and the aim is starved Lactobacillus GCR163 will use to investigate the physiology of slow growth and characterize the protein suppression in the presence of growth-limiting carbon source method. So we use mRS. mRS is special media to grow these kind of microorganism in it, with 0% lactose overnight. We culture them overnight and we inoculate them in three different different conditions. 0% lactose, mRS with 0% lactose, which is starvation, 0.2 lactose with growth limiting, and 1% lactose, no growth limiting, is our control. And we grow them under this condition, the growth condition. We use a buffer to buffer the media, which is 0.35 potassium phosphate buffer. And we start with the low initial optical density, which is 0.02, and we grow them aerobically, and for 30, and temperature is 30, and we sampling in day 1, 2, 4, 6, 8, 15, and 30. Harvest them. We measure the optical density, pH, bacterial viability, and dry weight. Also, we sample some sample for the SDS phage analysis and 1D LC-MS analysis. Result. So the growth curve of this bacteria in 0% lactose, as you see in the first graph on the top left, 0% lactose show high viability after 30 days compared with our control 1% or 0.2%. Then the other says, in contrast, then subsequently decrease in the cell density for 1% and 0.2% in day 6 and 8, which start dying, but 0% still surviving. pH declined, and this treatment reflects the OD, as we mentioned before on the top, indicate that the acid protection from lactose could affect the growth of Lactobacillus casei. SDS phage. The picture here it has some lines. Line from 1 to 5 is, I'm comparing 1% lactose, which is our control, 0%, which is the treatment, starvation. So lane 1 to lane 5 is 1% lactose, and lane 6 to lane 10 is 0% lactose, the bacterial growth from day 1 till day 8. And there is obvious difference between these two in the protein expression in these two treatments. I highlight some upregulated protein in 0% lactose, and other group of proteins that are downregulated in 0% lactose compared with 1% lactose reflect, or they could indicate they could be associated with lactose starvation. And the picture here is for SDS phage comparing 0.2 and 1%. There's not really obvious difference between these treatments. Before we run the samples for 1D LC-MS analysis, we run them on the gel again. We take the sample from stationary phase and run them on the gel, and the lane 1, lane 2 is 0% lactose, and lane 3 and lane 4 is 1% and 0.2%, and there's a difference expression between these treatment. 1D LC-MS. So before I go in detail on 1D LC-MS, we usually have the protein mixture. We lyse the cell, we have the protein mixture, and we give it to CSL to analysis the protein samples that we give them. So 1D LC-MS is the one-dimensional liquid chromatography mass spectrometry. So what they do, they inject our sample in the chromatography, and the chromatography will separate our proteins in the samples based on the protein hydrophobicity, and that will generate spectra, and the spectra refer to the proteins in the sample. So 4 months later, doing the analysis of the data, we feed back the proteins on their original pathway, and we apply some statistical method. We end up with a page like this, huge number of pathways, and the numbers here shows the only significant up or down of the proteins. Today, I'm going to talk about some of these pathways. I haven't finished actually the analysis of all the data, so I'm going to talk about galactose metabolism and pyruvate metabolism, which is related to carbohydrate metabolism pathway, and alanine asparagine metabolism, and purine metabolism, which is related to nucleotide pathway. In these graphs, I just want to show you something. I choose proteins related to galactose metabolism. On the top left, the blue column shows the proteins are upregulated in 1% lactose compared with 0% lactose. So on 1 is upregulated and 0 is really downregulated. Other group of protein related to arginine metabolism, pyruvate metabolism, they are really upregulated in 0 but down in 1. So what's happening in details? The picture here shows outside the bacteria and inside the bacteria. So when the lactose is present in the media, there is a special receptor on the cell wall of the bacteria called phosphate transferase system, which phosphorylate the lactose and make lactose phosphate, and by a special enzyme like phosphate beta characterise transfer to galactose phosphate, and then we end with the pyruvate. Then from pyruvate they make lactic acid or different end products. So in 1% lactose, we found these products are upregulated, or these enzymes upregulated, while in 0% lactose, most of the enzymes We found these enzymes as well, but they are down-regulated. But one more thing is the pyruvate is up. So what's happening? Other component in the media, which is amino acid, like asparagine, the special receptors in the bacteria cell wall take the asparagine or any amino acids, convert it to the aspartic acid, then aspartic acid to oxalic acid, then pyruvate, then make energy. We are not sure yet if they are they using this amino acid to make energy to survive or to survive and multiply. So 1% lactose, as you see in the top, we detect these proteins or these enzymes, but they are not really significant, but there. But in 0% lactose, they are using this enzyme are really significantly upregulated compared with our control. Purine metabolism. I'm not going to go into details of the pathway. It's really terrible and big one, but this is the actual pathway that I'm really working with them. So purine metabolism is one of the pathways related to DNA and RNA synthesis. So what's happening in 1%? I just want to say that in 1% we detect most of the protein in this pathway, and they are really aggregated. So the bacteria has the lactose, they use the lactose to make energy, and they divide and they grow happily. In 0%, most of these enzymes detected as well, but they are really down-regulated. So conclusion, our strain GCR163 survived for 30 days with a low and no obvious carbon source. This suggests this strain was able to find alternative pathway or alternative energy, such as amino acids, in the modified mRS. The SDS page identified upregulated protein and downregulated of other group of protein could associated with the lactose lactose starvation. When the LCMS results suggest that starving Lactobacillus induce several proteins related to amino acid metabolism, possibly to supply cellular energy. Bacteria usually adapt to nutrition stress by adapting physiology state characteristics, by downregulation of nucleotide and protein synthesis, and simulate upregulation of protein degradation and amino acid synthesis. Finally, I want to just thank my supervisor, Margaret Fritz, John Bowman, Ralph Nelson, and I also want to thank Dr. Richard Wilson from CSL Center for running my sample. Also, not forgetting my friend Jay Kacharantun to help me announcing the data. Thank you.